So welcome to the session on how selection decisions are made in dynamical field theory. And I'll just begin by recalling from last lecture the attractor states of the neurodynamics that we saw, the self-stabilized peaks that are instances of representation, are the units of representation we're using, versus the sub-threshold activation patterns that don't impact on anything else. So they are sort of off states. And we looked at how the self-stabilized peaks arise from the sub-threshold activation patterns in the detection instability. They arise, I'll say more about that in a moment, and disappear again in the reverse detection instability. And I showed you the capacity for fields to make selection decisions in the simulations very briefly, and I expand on that a lot today. And I also mentioned that there are these working memory states in the fields, and I'll be talking about that next week quite a bit. And there's a typo here, it's supposed to be boost-driven detection. I also mentioned that. And so last week we elaborated on detection instability. It shows, or it's the property of dynamical fields that even just responding to an input in these fields amounts to a decision. So it's a decision in the sense that the state before that decision, which would be sort of the off subthreshold state, becomes unstable, and the system then switches to an alternative state, the on state of the system. So this discontinuous change is how decisions are represented in DFT, and that's actually very important. I argued because that makes the decision macroscopic. It avoids the criterion that is used, like a threshold that is used to really enforce the decision. And actually, at the end of the sequence, you will see that we will have a look at how decisions have consequences, and that is connected to the fact that they are represented by macroscopically different patterns of activation, not microscopically different patterns of activation that are just categorized from the outside. So the self-stabilization of the decisions, of detection decisions, is operationalized by the neurodynamics being bistable, so having a on state, a peak state that overlaps with the off state, and to switch into the on state when that bistable regime ends, becomes monostable on, and it is limited on the other side when the self-stabilized state becomes unstable and you only have the off state left. The other side effect of the detection decisions is that they are responsible for the emergence of discrete events, events at discrete times. And in the later parts, which we'll discuss early next year, in January, we'll discuss how sequences of mental states that can do something like thinking and inference and so on, how they arise from sequences of such instabilities. And last week, we also discussed in some detail empirical evidence for the detection instability from perceptual hysteresis. Here's a reminder of that figure that refers to the detection of motion. I also talked about new data about the detection just of contrast. And I mentioned that there's not a lot of evidence for this. It's very difficult to study and maybe often considered maybe too polluted by non-perceptual factors. But very careful analysis has in the meantime established firmly that there is hysteresis in very elementary forms of perception. And that will be consistent with this idea that the transition from off to on is not just a question of applying input, and a graded output is suddenly judged to be above threshold, but there is really this bistable regime. So today I want to talk about the selection mechanism and the selection instability. I remind you in this figure of what that's about. We're talking about selection with respect to multiple possible values along the dimension that is represented by a field. Here indicated by the fact that input has two localized, two locations with maximal input. So it consists of two localized contributions, so that you could think of either this location or that location here at the bottom, specifying the value along that dimension that you want to select. And if we enforce a field to have only one peak in the field through enough inhibition, let's say global or long range inhibition, then we can realize the situation where you can have a peak either over this location or a peak over that location. You see that the other location is inhibited by the global inhibition here from this peak. And so these two graphs here at the bottom and the top demonstrate the bistability. In this case, it could be multi-stability because there could be many local maxima of input. And this bistable regime has potentially a limit, a boundary where it turns into monostable. For instance, I'm indicating here in this middle panel that if the difference in input strength between these two localized inputs is large enough, then maybe only the peak centered on the right is viable. The peak centered on the left would have activation here being pushed high enough to go through detection and then suppress the other peak. So that will be a monostable regime. Of course, symmetrically, the other monostable regime would also exist if the discrepancy in input strength was symmetrically opposite. And generally, there can be, this could be multimodal, and there are many different ways of how you could make selection decisions more and more unique by making the inputs less and less equal or homogeneous. So that's the mathematical principle that we looked at in live simulation. And so today I want to talk a little bit about what that means. So first, functionally, what it means is, again, the same idea that the bistability of that decision stabilizes decisions, which are now selection decisions. So here on the right is just a simulation with a lot of noise of this kind of situation where there's some localized input at two locations here of the field. And in this case, the leftmost location was selected, fluctuates, and it really suppresses the rightmost location. You can see with the naked eye that this is a macroscopic difference in activation between the two choices. And that makes the possibility that by chance, by fluctuation, you would switch to the other choice, make that very rare, very little, very unlikely. While here on the left, just illustrating, all I did here was put this surface through activation at some level, just to visually highlight that if we are this just input with noise, that if we were to just to take something like pick the local maximum or look for piercing of a threshold, then under reasonable assumptions, more than one location could be above that threshold, or the location at which activation is maximal could vary in time as inputs fluctuate, especially if these inputs are relatively low level from sensory surface. This would be not implausible. And so here you would say the selection decision isn't stabilized. You would maybe select this location, but then jump to this one and then jump back and so on. There would be multiple switches here in your choice. And that is not a good thing. If you're a motor system, you change your opinion like that. We once built a robot model. I'll actually refer to that like 2 lectures down from here. That has to make a selection decision like that. And for some historical reasons, we actually emulated something like this here, which was a real mistake. And we did actually, when we did run the robot, we actually observed that the robot was not making the selection decision. It was a switching back and forth and meandering between the two targets. And in the robot, there was a real need to link the robot continuously to its input. And that's how we saw that. While, of course, in many models, you're just looking when threshold is first pierced. And then from then on, you're running some other model and no longer taking into account the neurodynamics, which is not a realistic picture of what happens in the nervous system. And this sort of algorithmic way of stabilizing decision is actually what is routinely done in engineering, where you have some often probabilistic decision-making algorithm. And once you've made the decision, you're not actually going to look back to that input channel until some finite time later, which is sort of the earliest time when you would allow a revision of the decision. So functionally, stabilization of selection decisions is important for linking these kind of cognitive levels that can make decisions to noisy sensory inputs. It's quite critical. See in a couple of examples, see these intuitively. Now, how do you actually see empirical signatures of this sort of mechanism. If you think about selection, you could think everything we do all the time, every day, is a selection decision, because all the time you're selecting what to attend to, maybe what behavior to activate next, what thought to allow to come into the foreground, and so on. And so you could think of cognitive life consisting of a sequence of selection decisions. And so it's very, very important, very fundamental. And so how can we get some sense that this form of description of that makes sense? Now, it turns out that the literature on that is actually a little complex, and it's complex because it's difficult to study experimentally something like free choice, just letting you decide what you want to do. In contrast, most experiments actually have what we call an imperative signal that tells the participant what would be the right choice, what choice to make. So that the choice would be reproducible from trial to trial and from participant to participant, except the participant makes an error, doesn't do the right thing. You could say that when these errors become very frequent, then you approach something like a free choice situation. The reasons why that is really the way things are studied have to do primarily with experimental issues, and reflect perhaps why it's difficult to just predict and experimentally describe how people behave in daily life when they make seemingly free choices. I'm not talking about some deep philosophical thing of free choice. I'm just saying, where people make these choices all the time, where there are multiple possible outcomes that they would actually sometimes realize. So it's difficult to do that with daily life sort of situations. And the reason for that is ultimately that there are too many other factors than the ones that you can experimentally control that will contribute to these choices. So when you put people into a experimental situation where you don't tell them what to do, so they have task uncertainty, they will very often adopt strategies that can then be individually different, that they might persistently use and that destroy the free choice that you apparently had. So for instance, when you ask people to respond to a stimuli in some way, you don't tell them which button to press, or which target to look at, or whatever your paradigm, then they will find a simple thing like the one that's closest to your starting button, or the one on the boundary, or the largest one, or things like that, and then can stick to a strategy like that. And that's not very informative. And I'll discuss later actually the task set, so how things are distributed and what kind of expectations you have about an upcoming task do play a major role in these kind of studies. And I'll be just in the second half of this, I will be discussing that in detail. There are a few cases in which one can study choice without an imperative signal, and they're all based on things happening very fast or very automatically, so that you don't really intervene. And I will elaborate here on the most prominent paradigm for that, which is selection of a saccadic location. And 2 lectures down, I will be talking about, or maybe next week already, talking about the other famous case in which you use babies. Babies don't use strategies in some sense. They very reliably react when you leave them free to choose in a very simplified situation. We'll see the A not B phenomenon we'll study as an example of selection decisions. So saccadic location, this is the decision where to direct your gaze. You have to know in the background that voluntary gaze shifts are so-called saccades, that is abrupt, somewhat stereotypical eye movements that land you on a new target in the visual array, that they have very uniform trajectories that are just dependent on the distance to cover and so on. You can vary a little bit their profile, but not much. Well understood neurally, actually, how the movements themselves are generated. And you do them all the time. You're not aware of them. You do about 3 per second. So every about 300 milliseconds or something like that, you're shifting your gaze to a new location. These are actually the only eye movements that you can voluntarily control. You can essentially fixate, which means not making eye movement, although fixate can amount to an eye movement. If you move your head around or the object, then you're tracking that object a little bit. You can make pursuit movements, which is sort of like the fast form of tracking. That is, if the object moves so fast that you would lose it if you were to just fixate on it. And these pursuit movements can only happen. So the involuntary, they can only happen if there is a moving thing to pursue. So you cannot voluntarily make a graded eye movement across the scene. You have to have a visual structure there. You can all track my finger, for instance, when I move over the screen here, but you cannot make the same movement without a stimulus. So without a direct guidance stimulus, you can only make these saccadic eye movements. And they're quite fundamental. They are quite mind-boggling if you think about that, because really imply that every 300 milliseconds, the visual scene that's transmitted through your brain on the retina is shifting widely. You're always seeing these different views of camera. Just think of a camera that you would be moving around like that. That would be really very confusing. But that happens all the time in the brain. And so you're choosing where to look at every moment in time. So you're making every 300 milliseconds. It's not periodic, there are different durations, but on average, every, let's say 300 milliseconds, you're making a selection decision where to move next. While you're making this card, visual information is not registered as percept. So you're blind, as we say, during the saccade, and then you're picking up the image at the new location, working on that, and then you do the next saccade. There is also something called microsaccades. So there's some very little eye movements, sort of drifty, but jumpy little eye movements around fixation, which are also not voluntarily controlled, and that might serve some purpose that's still not very well understood, perhaps to induce more neural firing, because the early parts of the visual array are really transient detectors and react primarily to changing visual input. And I'll not talk about that. I'll just assume that fixation just means that you're looking at the same location for an extended amount of time. So that's a fast and involuntary system where one can reliably control parameters to elicit different patterns of looking, and I'll show you that. I would say there's a large literature on trying to look at where people, to analyze where people look at in natural scenes. So this is from one of those studies. And yes, people like to look at faces. They look in faces at certain locations, eyes, mouth, and so on. Also look at hands, but also at other objects. There's a long history of trying to find some universal laws in this, but that's not so easy. We know that where you look depends on the visual contrast. It's much less likely that you look into some part of the visual array where there's little contrast. You wouldn't look like here or into the white here and so on. So you tend to look at locations where there is a lot of local contrast, but where you look is also biased by top level stuff, like you're interested in people in general. We're very interested in social signals to understand if people are friendly or maybe dangerous to us. But it might also depend on tasks that you have, looking for an object, or looking for a place where you could escape, or whatever it is. And so that's a literature that is more descriptive in nature, while the precise mechanistic account for how this works takes radically simplified visual arrays. So the classical thing is you're in a dark room, there's a computer screen, you have a cross on the computer screen that you're supposed to look at. There's nothing here on the right. And then at some moment in time, a light shows up, like only this red light that maybe shows up on the screen. When that happens, you can actually barely prevent yourself from making a saccadic shift to that new location because of the transient that is induced there. If you're instructed to make a saccade, it's very easy to follow that. If I can tell you, if at the same time this cross is deleted as this light shows up, it is almost impossible to not make the saccade. You're really driven very strongly. These are called visually driven saccades for that reason. So when a single spot occurs, it's also called single step saccade, because you can think of this as a step, like this goes off and that goes on. that's used to study the exact dynamics of saccades and the latencies. And then the complex cases when two things light up at the same time. For instance, these two targets light up at the same time. And then you can make either a selection decision as indicated here. So you go to either one location or the other location, or you can make an averaging decision. Averaging would land somewhere in the middle of these two elements of contrast. The transition between those is a function of how far the targets are from each other. In fact, it happens pretty much, the transition from bimodal to monomodal happens pretty much when the two objects are close enough so that they can both be foveated. The fovea is this particular region, about 2 degrees visual angle, that you can, where you have higher resolution on the retina, where you have most of your finer perception. And so when these are closer than 2 degrees visual angle, you will foveate both by looking in the middle. So functionally, it makes a lot of sense. This is the sort of thing that you do when you do reading, for instance. You're not looking at every letter in the word. You're finding a location within the word where you can foveate almost all or a good part of the word. And it's not exactly in the middle of the word. It depends on the reading direction. So that also you can well studied. So for instance, in Hebrew, people are fixating somewhere else, like in the mirror symmetric situation than in Latin, for instance, or our languages, Latin languages. Now here is in the lab, there's a subtlety also that you could have a task where you are free to look. So you're actually not even told to look, but you're just going to do that automatically. Or you could have a task where you have an instruction to look at the red target and not at the green target. And there could be some way of how it doesn't have to be color, some cue of how to distinguish between these two targets. And this paper, actually, from which this data comes, is one that I'll refer back to that in a moment. Is one where people were looking at instructions like that, and one observation is that if you arrange things so that people look pretty quickly, that might mean, for instance, that this year the fixation light is turned off, then they tend to make very fast saccades. That under those circumstances, they might not actually be able to follow that instruction. They will sometimes go to the right target, but sometimes also go to the opposite target. And I'll come to that in a moment. So this is actually how we started our dynamical field theory saga, because we try to account for this elementary form of selection decision-making. You see, this regime here is easy to understand given what I showed you, because you could put the field easily into this regime where there are two localized inputs corresponding to these stimuli, and then you can generate a peak either over one or over the other location, and it's bistable. And then you would interpret the output of that system to be the saccade. So in this early model, we just had the field representation. We didn't actually model the eye movement. Since then, we've done more complete models that also deal with that. Initially, not so obvious cases where you have this averaging response. So there could be two sources of averaging. Either your input is smeared out so that the input has a local maximum. It's not really plausible for these because you do actually see these different stimuli. It is, of course, we don't know if the structures that control saccadic movement generation, in this case, frontal eye fields and the colliculus superior, or in English they say superior colliculus, if they have that kind of resolution. But there are other data that suggest that this is not just on the input side. So for instance, when one of those choices occurs much more often than the other in a single target situation, and then you combine them, then the center of mass of this peak is shifted toward the more frequent location. That's an effect we modeled. And that's an effect that requires us to assume that the input isn't monomodal, that it is actually still bimodal, and then the averaging is an active property of the field that computes the motor command in a sense. And what happens in the model is that when the localized inputs are close enough to each other, then the excitatory interaction can fill in the interaction. So a peak energy on the center can become stable because the activation on this border accurates the right side and vice versa, and the excitatory interaction can then lead to averaging. That's the account we provided. Now, if you, so this just summarizes what I just said. Now, if you look into more detail, the most interesting detail for this kind of theory, of course, is what's the time course of saccadic selection? So how long does it take? If you leave different amounts of time, what would be the trajectory of decision making? That has interesting structure. You see, it's all data. We modeled literature data at the time. This was all known, but maybe not interpreted interestingly. And so that's what we're trying to understand. So in this old data, you see on the horizontal axis here, the time to saccadic onset. So the time between presentation of the stimulus and initiation of the saccade, onset latency. And let's say, just to make things clear, this would be where a single target is flashed. This is the target. This is the matrix of the saccade, the angular location in angular coordinates where you're going. And so different latencies in a single target, well, you just do this saccade in the right location. These are two different participants, no problem. Now, if you have two targets, one of which is marked as target and the other is marked through some feature value as non-target, then you see the situation is a bit more complicated. You get some saccades that go to the right target, but some also that go to the non-target and some that fall in between. If you look at the finer structure, you see there are crosses here and there are open circles. And the crosses were trials where participants were instructed to make the saccadic decision as fast as possible, while the open ones were conditions where they were asked to be as precise as possible. And this way, participants can modulate their saccadic latency a little bit. You see these crosses tend to be here at lower saccadic times. 200 milliseconds is really fast. There are some very fast saccades here, which we call express saccades that are like 150 milliseconds. And then you see the open circles, they overlap, but they tend to be at somewhat larger times here. And you can really see with the naked eye where the these late data are all on the right target, while the early data sometimes do averaging. So there's a temporal transition from averaging to making the correct decision in this example. This can be formalized. You can look at how much averaging there is in the data and compute it in some way, and you see that that averaging decreases over time for both participants. There's a lot more data in this paper and this was studied quite intensely for a while. So to understand that theoretically, it turns out that that the model that I just sketched is not sufficient to understand that. And it's an interesting extension of the theory. That's why I go into it. So in the model that we've looked at so far, the peaks are stabilized by local excitatory and global inhibitory interaction. That makes them attractors. But I already hinted at earlier that this violates a principle of neurophysiology in the brain that we theoreticians like to call Dale's law, after someone who first postulated that. And that principle says that every neuron only forms one type of synapse on the neurons onto which it projects. And by one type of synapse, we mean in terms of the neurotransmitter, and that means it is either excitatory or inhibitory. You don't have a neuron that makes some excitatory synapses and some inhibitory synapses. This is why we even talk about excitatory neurons and inhibitory neurons, because that's just short for what kind of synapses these neurons make on their targets. So you see that the model as written down is not inconsistent with that, because if we think of locations in the field as neurons, which we know isn't actually quite correct, but just for simplification, then the neuron here would have excitatory synapses here on its neighbors, and you would have inhibitory synapses on the dendritic tree of neurons that are further down the stream. So this is not consistent. To repair that, we have to essentially introduce inhibitory interneurons. I believe I mentioned this before, I don't remember exactly. Inhibitory interneurons would be neurons that are inhibitory in nature, that is, the neurons in this field project onto, here in this case, back onto the excitatory layer with inhibitory synapses, so making negative inputs to this layer. But the inhibitory neurons would be driven by excitatory neurons. So the special architecture that is common in the brain, it's actually, you can actually analyze these cortical layers and they have a structure like that, is one in which these inhibitory neurons are not driven directly by afferent input from, let's say, from the LGN or from the retina, but they're driven by neurons that are excitatory in nature that receive that input and then pass it on to the inhibitory layer. So some external input would come into the excitatory layer, would excite the inhibitory layer, which then in turn inhibits the excitatory layer. So that's the more general account for how the kernel comes about. So you would say the excitatory part comes from connections within the field, while the inhibitory part goes through this extra synapse, first drives up the inhibition, and then the inhibition returns. And all of these have kernels, that is, they depend on how far from a location you're looking. So there would be some spread from the peak to the inhibitory field and some spread on the way back. And that is actually the reason why inhibition would generally be spread more broadly than excitation, really just because it applies a kernel twice, and that makes the effectively spreads the inhibition. So chapter 3 of the book, of the DFT book, that was background reading for the exercise that's discussed today that you handed in last week, actually discusses two-layer fields in some detail. You can go back to, there's a box there about that, and you can go back to that, and I'll show you the simulator for that in a moment. Just make it concrete, in our description, these are both still Amari fields in the sense that the sigmoid is inside the integral, so it's u dot equals minus u plus h plus s, and then the integral is, this would be the familiar field, like the Amari field was looked at so far, except that this is only an excitatory kernel now. All of these kernels are just simple Gaussians, so they don't have these two components. And it gets inhibition by a projection from the other field, the field V, which will be the inhibitory one. So this negative kernel here, the kernel is positive, and I put the minus sign in front of it. So this would be the inhibitory contribution. And the inhibitory layer would be a simple, low pass filter, this first part here. And all it has is input from the activation variable itself, the U variable. That's the mathematics. So it's not very mysterious, even though initially it looks like a really large equation. I wanted to show that to you by going to a simulator. This is a simulator we don't have online. It is in the Cosivina package. So if you look at the dynamicfield.org webpage, there is a pointer there to the, sorry, I didn't want to, actually this I want to read later. So here you can go to software, and under software there is the Cosivina package, and maybe some of you already looked at that. You can also use the CDAP package, by the way. It's a little different in use. If you use the Cosivina package and you have access to MATLAB, as you should have as a student when you're in Germany, is actually a student allowance for almost all universities in Germany now, a student version. Then you can do all the things that I do by just following the instructions in the Cosivina, on the Cosivina webpage, which I don't want to do now. And then, so what I have here, use a sub routine, an example routine. That's the launcher 2-field layer preset is in there. And if you run that, you get a simulator, somewhat like the simulator you're already familiar with. It has, now it doesn't show the kernel, it has these two fields, the U field and the V field. Quite a bit of noise here in the default setting. This one has options to select parameters that you can preload, and it just means these buttons are already given some meaningful values. I think I want to reduce the noise just a little bit here. Not sure why there's so much noise in the default setting. And And so now this is the excitatory field, this is the inhibitory field. And again, I can put external inputs. These inputs go into the excitatory field. So you see the green line is the excitatory field, right? You see it tracks that. And then I can go through detection. It has the same dynamics in that respect. It goes through detection, the peak builds, and that peak, that suprathreshold activation, then drives up inhibition. The red line, again, is the sigmoid applied to the inhibition. And that peak then can inhibit, induces this inhibition there. I'll go through that again just to make clear. Sort of you see a sequence of events where as red arises on top, now it takes a moment for red to arise at the bottom, right? And once red arises at the bottom, let's again go back. Once red arises at the bottom, then watch how inhibition, how it suppresses the field elsewhere. So when I induce the peak, only once the red peak at bottom has appeared does the inhibition show up. See? Yeah, that's the point I want to make. So this can also do selection. This is exactly what I'm talking about, right? And so it has a detection instability, so I can, for instance, switch it around this way, right? This is the thing that we want to demonstrate. That's all I want to show you right now with this simulator. Oops, wrong button. Here, no, where is it here? So I introduced this generalization because it has observable consequences, and the consequences that there is some natural sequence of events in two layers fields like that. It makes intuitive sense. So, and the first thing that will happen is that you will get activation just in response to input, because the first thing is that the system reacts to that in a sub-threshold, sub-threshold solution. And then once excitatory layer has reached threshold, so that now the sigmoid starts to return values, excitatory interaction will start to have an effect. I'll show you in a moment that leads to averaging. And only once inhibition has reached threshold. Now, inhibition will be driven by excitation only once the excitatory level goes to the threshold. So only so that drives inhibition up, and only once inhibition has itself reached the threshold can its effect actually be seen. And here are examples from our model of this time course of decision making. This was published a long time ago. You can see these different effects. So I'm modeling the two locations, saccadic selection task, activation over position here of the endpoint of a saccade. And here, this little dashed line, that's the stimulus reflecting that bimodal two locations are stimulated. And you see that the field 10 milliseconds after this input was derived, the field is sub-threshold. So the field is still at resting level and has some local peaks that are just mirroring that input. So that's just input, low pass filtered version of the input. When you look a little later, like 20 milliseconds, the peak have grown a lot, self-excitation sets in, they still reflect a little bit the bimodal structure of these inputs, but already you see the peaks are sort of pulled in, and at 80 milliseconds, excitatory interaction has engaged, the activation is positive now, and it has filled in the activation between these two peaks. So that's the account for modeling, for averaging. And if you wait longer, and this is shown here in a different time scale, these are and using this gray level plot. So it's the same X and then here time, and the gray level indicates suprathreshold activation. You see here early the bimodal regime, and then here the monomodal regime. And so at about 150 or so, the inhibition sets in and supports selection. So now you see that the system actually moves to the rightward location and makes the decision here, in this case, of the rightward location. And this model accounts for the data. This is from the example I just showed you. So for instance, here in the model, the latency was manipulated. It was manipulated here in the model in a particular way that I think I'm going to gloss over, having to do with the fixation system. So the correct choice here was the lower choice. And you see that here in the early data, you see averaging, and in the late data, you see that it does the correct thing. You see this transition from averaging to selection. This would be an example where you don't instruct on which target to select. So there's symmetrical inputs in the model, and as a result, the late distribution is bimodal. You can either select one or the other, but early on you will be doing selection. So it's interesting, that time course of selection can be accounted for by the architecture, by the coupling of excitation inhibition in the system. And that's a very generic thing. You're very much in the nervous system. You find that early on there tends to be averaging. People also talk about the system having a large scale, taking into account all inputs early on, and then being more selective and drawing back onto particular inputs later, and they do that by inhibition. And inhibition just takes a little time. It's important perhaps to realize that this is due to the architecture. It's not due to the inherent timescales. So for instance, the excitatory neurons are actually biophysically often considered as slower. That is, activation, the membranes are not as fast. The inhibitory neurons are smaller and have a smaller soma, and they can actually react more quickly, electrically speaking. So that they are coming in late is really just in terms of sequence of events. It's also not just the delay at the synapse that you have these 2 milliseconds until the inhibitory layer is reached. It's actually the reaching of the threshold that as a population you have to get enough of these inhibitory neurons going, and that means more than a certain subgroup of neurons needs to reach threshold, and only then will the inhibition be felt, and that is what accounts for this temporal ordering effect. Gloss over this one here. The other interesting thing is that these 2-layer fields afford oscillations, and I believe some of you already touched on that topic when you looked at the 2-neuron simulator, and you can play with that. There's an exercise associated with that you can actually do on the 2-neuron simulator on the web page. And here I'm not going to come back to that. It's a very generic mechanism. So in the brain, you very often have these dipoles of excitatory and inhibitory neurons that have this same coupling structure as I just hinted at, this coupling structure. And these systems have oscillations, so stable limit cycle oscillations under some conditions. They may also have active transients, or just a single event, a peak that then deletes itself, sort of like a spike, but it is a population level peak. And that is a source of time course generation in the brain that might be responsible for movement generation and for synchronization, for counting and things like that. So it's being used in a lot of places. It can also be pathological when these oscillations are globally coherent, then you have epileptic seizures. Okay, that is just this spontaneous decision making going for an imperative signal. So in the second part of this lecture, I want to talk about how selection decisions are studied in the laboratory with this imperative signal, not in this unstructured way. So there's a huge literature that goes back to the beginning of psychology. It was the beginning, reaction time is as old as psychology, Wundt, and I forgot some of the famous people who invented that now. And sort of its rough conceptual structure is always really has 3 things that are important. It has the imperative signal, which tells you what to do, which very often also is the go signal that tells you now you can start to act, but sometimes you can pull these apart. But then it also has the task set, which tells you what would have been different possibilities. So for instance, if a person has to, let's say, my little drawing here has to select either the glass or the book, let's say, that's the task set. So this person knows it's going to be one of these 2 choices. Maybe today it's on a computer screen, you have some buttons in front of it. So. And then the imperative signal would perhaps signify in some way the object to grasp in some form. Maybe it would have the word, glass, it would have some symbol that you have learned. And then you're invited to initiate your action as quick as possible. And reaction time is really the time between the presentation of the stimulus and the time when the movement is initiated. In the most classical paradigms, you actually had people initially being on some start button, and when the start button was released, then you measured the reaction time. The convenience was you didn't really have to measure the movement, which was much harder, and also didn't have to analyze the movement to just determine the reaction time. Sometimes people talk about response time as the time to the end of the movement when you achieve the outcome that was asked of you. That's not really a useful measure most times, because there is a time that the movement takes, and that time depends on things that have nothing to do with the initial selection decision. So today, most of us are not talking about that. And other people use the word response time and reaction time interchangeably, even though historically it has these two different meanings. So you see that in the structure of these kinds of paradigms, the task set is really important, right? Without the task set, the imperative signal doesn't really have meaning. And so we'll have to talk about that. So in many studies, people talk about the task set, for instance, as the nature and number and perhaps value of the different choices or alternatives of what you're supposed to do. So a lot of the initial work was about that. It was often about the probability of choices, which was connected to the notion of information in the Shannonian sense, which really the probability of an event. But people were also looking at some kind of degree of difficulty of recognizing or performing or selecting the choices. And I'll talk about a few of those. So often those were, for instance, about how intricate the code is. So if it's a more difficult mapping from the imperative signal to what it signifies in the task set, then that was supposed to require a lot of computation in your mind, and then you predicted larger reaction time. So it was used as a measure of the mental computations that are necessary to make the decision. And for instance, in the formalized tradition of mathematical psychology, in most elaborate, perhaps in cognitive architectures like ACT-R or SOAR, some of you might know that, reaction time is the primary measure that is a probe of your architecture, essentially asking if you're thinking of, if some processing is done in sequence or is done in parallel, that will make a big difference for how it scales with the number of items that you have to process. And looking at how reaction time depends on such parameters is then a tool to make measurements in some way on the architecture. I'll not go deeper into that. That's a whole field by itself. So from the point of view of our neurodynamics thinking, we often call the task set or represent the task set as a preshape of the underlying representation. And just it is shaped already, that information was available already before the imperative signal and go signal comes. So we assume the field has already relaxed to a state that reflects the knowledge about the task setting. So for instance, if you're selecting a particular movement parameter by moving toward some buttons on the computer screen, you could have let's say a 3-choice task, in that on that screen there are 3 buttons, and you already know it's going to be one of those 3 buttons, or you learned as a monkey across many trials that that's the 3 things that will change color and that you will have to point on to get reward. Then that would mean, in theoretical terms, that the neural activation field that we use to represent the motor intention is not flat initially, it is pre-shaped. Initially, meaning when the specific stimulus, the imperative stimulus arrives, it's pre-shaped. It would perhaps have some input at each of those locations that would perhaps average to create a certain kind of pre-shape. And the imperative signal would, when it specifies a particular choice, it would be centered on one of those 3 choices, let's say here the leftmost. And so when that arrives, it would drive the pre-shaped field toward a shape around the correct location, the leftmost in this case, but also actually push it through the threshold into a self-stabilized peak. You see, this here is a self-stabilized peak. It suppresses everything else. This is a sub-threshold peak. And so somewhere in here, the transition takes place. And if we have some model of the response unit, then it would be this kind of peak that drives the motor system. That's the account. And so this pre-shape reflecting task input is important. So there could be many different ways of how that pre-shape arrives. And next week, I will be actually talking about a way how it arrives automatically from learning, as we call it a memory trace. But it can also arrive through instruction. You can tell someone that there are these 5 buttons, but only the 3 center ones will ever go on. Then you pre-shape that based on instruction. And then, of course, the visual layout, the perception of what one would call affordances. If you have 5 objects on the table, you assume those are the 5 things that I might be asked to grasp. And you could already prepare your movements in part by pre-activating the corresponding motor representations, maybe also the corresponding perceptual representations for dedicating attention to those locations before you then actually make anything supra-threshold. So here's an illustration of how such pre-shaping could act. So I'll first look at the case where the pre-shape is a weak contribution. So here in time, you see there's a little bit of pre-shaping. There are 2 locations at which the system expects input, reflecting maybe this kind of input. And then an imperative signal would contain 2 aspects. It would contain a localized input, the different colors reflecting different trials, and where you would ask people to move to different locations. And then there could be a boost. So the whole system could be lifted up to some extent. And so the limit case I'll first be talking about is where the localized input is stronger than the boost. So the peak would be coming up, and shown here on the bottom is where the peak in the end will actually lie. So you see the pre-shape will influence a little bit the transient, but in the end you will make a peak over a location specified by the input. So that's the pre-shape as sort of a little prior helping you to find the right choice. So at the end of today's lecture, I will show you the opposite limit case when the input is a small bias and the pre-shape is the dominant term. That's when you have a categorical task, for instance. So in this limit case, you can try to account for all classical laws. So we did in a paper we wrote about 10 years ago. We accounted for classical facts. Maybe those who studied psychology know Hicks' law. It's like from the 50s. Hicks' law says that the reaction time and the choice reaction time task increases with the number of choices. It increases roughly like the logarithm of the choices. So if you double the choices, number of choices, there will be a graded linear increase. It's not quite true. It actually saturates at larger number of choices. And that can be accounted for in this theory in a subtle way. It's actually surprisingly subtle. Here is an example. We're always looking at the leftmost choice being made, and we added choices as different local pre-shape peaks that are all sub-threshold, right? They're just a little bit of a pre-shaping. And so there's no order to this. So we take the leftmost really just for simplicity, but the longer arrow means that that's where we'll get the specific input. And we keep track here in this image of the activation level and where the local maximum is in the end. So it's always over the leftmost here in this case. And you see that when we apply the stimulus, we apply it at 0, so it takes some time to grow. And you see here that when you have a single peak arises earlier than when you have 2 peaks, local inputs, or 4 or 8. So if we take some criterion level here for this activation as a measure of reaction time, when that level of activation is reached, you assume reaction time, then reaction time will increase. And this is actually from the model. This provides exactly the correct function. Don't take me wrong. It's not that the piercing of the threshold is the mechanism for reaction time. We're just making that measurement on that system. We're, of course, thinking as a mechanism in the brain that it's the peak that then drives the motor system that is really bringing about the reaction. And that peak is not really present from a threshold on. It would arise gradually and then would flip the motor system at some point. And it's this detection stability that makes that a relatively sharp event. But just as a measure, we took some threshold here to mark these times. Now, because it goes through the threshold steeply, that's not very sensitive on that. It's a subtle effect because this is a supra-threshold pattern, so it slows down. If you look at 8 choices, let's say, slows down the growth of the peak really just because the sigmoid is soft enough for these to have a slight inhibitory influence on the evolution of this peak here. And it's interesting that a parameter fit of a lot of different experiments, this was just one, always reveal that the parameters of a model like that describe human behavior are always such that below the detection instability, before you make the decision, the field is very close to the instability. So here, in the sense that the sigmoid is soft enough for this level of activation to already matter a little bit. And you could think of that as sort of a homeostasis process where you're tuning yourself to be sensitive to inputs rather than selecting very early. You kind of adjust your system to have to give every input a possible role. So we find that very often to be the right parametric regime. It's not the kind of regime we want to use in robotics where we would like to have very robust things, but it makes some functional sense that the nervous system is not trying to avoid to commit to single choice early on. Thus, it might be tuning things so that being close to the threshold means every choice gets a little bit of a vote. I'll come back to this law in the form of the Hyman law in a moment in a different context. This is just one representative of a large number of typical experiments people did at the time that we all modeled in this paper, if you ever want to look that up. Now, more interesting to us are metric effects. These are effects that are not typically studied in cognitive psychology, were not at least at the time. And we're predicting something a little bit counterintuitively. We're predicting that when choices are closer to each other, then they will be faster. You might intuitively think if they're closer, then they will be slower because it's harder to discriminate between the two choices. And I'll show you that that effect that's called distance effect occurs under different conditions. I'll show you that at the end of today's lecture. So the metric effect is the opposite of that. You see here where it comes from. This is just the preshape in the field, irrespective of the specific input, the imperative signal. And so when you have two choices that are far from each other, then we're assuming they both get some input at these two locations. Let's say there is some perceptual structure that tells you which movement direction that is. And you know, when you bring up a peak, let's say the rightmost, then you will do it from the pre-shaped level. And the narrow condition here is just the same two inputs just put closer to each other, so they overlap. And the fact that they overlap makes that the pre-activation at this location gets a boost from the pre-activation at that location. That contribution hasn't decayed fully, and that lifts activation up here a little bit. And that makes this difference. That makes that when you have a close neighbor, you start at a higher level and therefore you reach threshold faster. It's only possible because it's still bimodal, so you're still able to really select the right mode by the specific input, which boosts one of these two locations. This is just a preshape that's symmetrical. So if it was monomodal, then you couldn't actually make the right selection. So this is a certain dynamic regime in which we're looking. Now, this is empirically true. So this has a very interesting history in that this was, we did this, like late 90s. I moved to Marseille at the time, where a lab where someone called Jean Requin had a long history in doing experiments like this, and was doing this on monkeys. And I worked with him and Alex Sarila on studying the neurophysiology of these kind of decisions. I talked about that a little bit last week. And interesting story is that this question, this issue of the metrics of choices was something that he had studied earlier, like 10 years earlier, and he had not been successful in publishing it very well. It ended up in a French journal. It was actually an excellent experiment that had a lot of interesting controls in it. And it was published in French in a French journal, Le Travail Humain. It's not exactly a top journal, certainly not for these kind of psychology questions. And the reason was that people were not very interested in this because they were really thinking in information processing terms and the amount of information, probability, number of choices, and those were the things they were interested in. And the metrics for choices seems like not an interesting question. And in this theory, of course, it's interesting. It's connected directly to neurophysiology because, as I'll show you a little later, we can actually directly link it will be actually next week, directly link the metrics of these distributions to something in the brain, to how broad the peaks are in the brain. And we can have behavioral measures on that. And so the fact that the metrics matters is really sort of a signature of the neural mechanisms underlying these choices, rather than information processing is more an abstract picture describing what computations are made without specifying the neural mechanism. So long story short, Jean Requin had a published data set that I haven't reproduced here now, in which he had observed this metric effect, but it wasn't otherwise known in the literature. And we then much later reproduced these experiments and expanded them a little bit. So just to give you an idea, it's people make a center out task. You have a home button, you have a stylus on the home button, and then different ways of how to indicate the choices, for instance, with arrows or with some target, and different variants of this. And people, it's a very simple, small movement. And we're looking when they lift off the home button to make the movement. We did this with 2 or 3 choices and so on, but the main point was the angular distance between the choices, which we varied here. And we looked at response time, and you see the metric effect in the sense when the choices are further from each other, you have a longer reaction time than when they're closer. And actually the transition happens around 60 to 100. The 90 degrees has some symmetry that makes it perceptually a bit different. That falls, there's a little bit of inversion, but it's still true overall. When you do these kind of experiments, you have to look into all kinds of subtleties. For instance, can you foveate all? Are the choices just slower because you cannot foveate on the different targets in the same way? So you have to make sure that the way you present the choices is attentionally simple, and there's a control for checking that from where you just have to detect something at those locations. And then where do you present the imperative signal? Do you do that also in a location that you can foveate at the same time as you foveate these choices? So stuff like that has to be taken care of. I want to show you a more powerful and better published study that uses a statistical interaction effect, which for some theoretical reasons, some of you might know, is a better measure of such things. And that study takes the Hyman version of the Higg effect as its basis. So the Hyman's law is the law that the more probable a choice, the faster your reaction time. And it's really true in terms of the probability, the logarithmic logarithms is the logarithm of the number of choices is the probability. It really makes that uses that concretely, that form. So here on top you see how the Hyman effect is modeled in the model. So if I have 2 choices, let's say, and the preshape between those is different. This is a higher preshape than that, and we would essentially reflect the probability in how high that difference is. So if there's a larger difference in preshape between this choice and that choice, it would be higher difference in probability, and this would be a lower difference in probability. And this can be supported by the memory trace that I'll be showing you. So we proposed a learning mechanism of how the preshape emerges from that, and then when you have more trials relative to the other one, the dynamics will drive activation higher at the more frequent location than at the other location. And so when you have that difference in preshape, you get the difference in rise time. So again, it's always the maximal activation in the field as a function of time. And then you see that the high probability choice rises earlier than the low probability by a larger margin when the difference is larger than when the difference is smaller. Now here we're looking at statistical interaction in the sense we're saying, what about that same difference in probability difference same, but now with different metrics. Now they are metrically close. Here they were metrically far. So you see when they're metrically close, the 2 pre-shapes overlap, you get a monomodal distribution, but it's centered here on the right choice. The more probable choice is larger vote. Its peak is larger, and the less probable choice is here in the flank of this peak. And lo and behold, you see that now the probability effect is gone. So the difference between the low and the high probability in this case is just the same as when the difference in probability was much weaker. Now we have the same probability difference here, but the effect is like when the probability difference was weaker. And that's just caused by the neighboring choice. Essentially what happens here is that the low probability choice gets a boost from the high probability choice. So it's lifted up here from the neighbor, and that makes that choice faster. And it's the low probability choice that becomes faster rather than the high probability choice becoming slower. So it shifted here to the left, and you can see that if you compare this this to that, you see where that lies. So this is an experiment that we published also a long time ago. We did other kinds of things in this paper as well, but one thing was that we looked at, again, center out tasks. The not so frequent, rare target was always the same across different conditions. For instance, here the vertical movement. And then there was another choice that was either a far movement direction or a close movement direction. And that was the frequent target. That was, I think, maybe 10 times more often, occurred 10 times more often than the rare target. And so in the wide condition, you see that reaction time has the predicted Hyman effect. So the rare target takes longer to prepare than the frequent target, while in the rare conditions, the same movement that we're looking at. It's always the rare condition that we're looking at. And you see now they have the narrow, the rare and the frequent have the same reaction time. And they have the reaction time that's faster. They are closer to the frequent target. These gray bars show one of the controls where we just have a simple response time where you just have to pick up the stimulus to test for these attention effects. And we also did ERPs, evoke potentials, and there's some story about that. So this is, of course, an experiment. You know, we had all the symmetrical conditions on the left and the right, and we varied which target was and so on to get to equalize over all those other factors that could affect reaction time. So direct effect for the support for this metric effect that is maybe somewhat counterintuitive. I think I want to skip over that. That's just some combination of the detection and selection, and skip over that. And the last thing I really want to explain today is how this magic effect can be turned upside down. It could become the distance effect, which is in the literature, it's much older. And it turns out that has to do with the other form of making detection and selection decisions, namely the boost-driven one. Remember, we did in the simulation this notion that when you only have a go signal and you don't get localized input or very minimal localized input, that then the preshape, the prior structure of the field will primarily determine which choice you will make. And it's under those conditions that we get the opposite magic effect. So I don't have to repeat that here. So this illustrates the same mathematics, but just quantitative difference. So now we're looking at a field where the preshape is sizable, this sizable preshape, and the specific input, the imperative signal, is just a small localized input, so smaller than the preshape, but a sizable boost, so all choices getting activated, so that the combination of this localized input, the boost and the preshape, makes that you go through detection threshold. And what you see happens in this case is that the peaks under these different specific inputs all line up, so they're a little bit pulled apart, but most of it is really determined by preshape. So you would say this field responds categorically. It just always presses the right button or the left button. And these different inputs, we could think of inputs that are to a larger or smaller degree associated with the right button. Maybe the blue one, for instance, would be somewhat associated with the right button, somewhat with the left button, if you want to think of it that way. And that will be just a minor contribution. So it's just a quantitative difference between the two paradigms. We need the boost to still understand how initiation takes place. If you didn't have the boost, there would be no guarantee that this local input will put you above threshold. So the more realistic picture is like this, where you do get a specific input that overlaps somewhat with one of the pre-shapes. So this little input here, and you get a boost at the same time, and then you will be realizing a choice that is largely centered on the pre-shape. It's not centered here on this stimulus, even though there's a little side trace of that. Now that we call that categorical responding. That is, now you're responding as dictated by pre-shape, and that will be a discrete set of locations. So when you do that, you get the distance effect. The distance effect is well known in the literature from the 1930s on. I believe, I forgot who the name of the person who first reported it. So a typical task for that would be that you have two objects that differ slightly, and you have to say, either if they're the same or they're different, or you might have to pick out one by some criterion. For instance, in the original study, you got two sticks presented to you, and you have to say which one is the larger one. And maybe you can't touch them, just look at them. And in that, the response time, reaction time to that is the longer, the more similar in length the two sticks, right? Makes a lot of sense. And this kind of experiment has been done in all kinds of variations, always ends up being of that nature. Notice that the response is categorical necessarily, because you have to say which is larger, which is small. You can't say anything in between. And in fact, it's all dictated by pre-shape, because there is no, if you were to be given a different stick that doesn't really respond to any of the things you learned so far, you can't make a new response. You can always just say larger or smaller. So it's necessary to take that picture that it's dictated by the pre-shape. And then the interesting thing is that then if you do it that way, you find that the magic effect is inverted. I'll say in a moment what that means parametrically, where you are. So here I plotted the same picture. There's a little bit of a difference that I'll point out in a moment. So here on the left, you have the choices have the same metric distance, and they have different probability. And I believe this has a larger difference in probability than that. That's the difference. You see this a little higher than it is here. And that makes here that the less probable choice, being the dashed blue one, takes a way longer than the more probable choice here. And that difference becomes smaller when the difference in probability becomes smaller. I think I misspoke. You'll see this is larger difference in probability and smaller difference in probability. I have to compare the pre-shapes. Here the pre-shapes differ less than they differ here. Larger difference in response time, smaller. Now, you can also look at error rates, but let me ignore that for a moment. Now here on the right, we have the same probability distribution. So it is actually the smaller difference in probability that is maintained here. And all we did is push the choices closer to each other, magically closer. Now, difference is, it's really from the pre-shape that you have to make the decision. You're going to make a decision in either one or the other pre-shape location. It's not, the imperative stimulus is not strong enough to just induce any peak. And so we're always in the regime where they are competitive, where the choices are far enough from each other so that they inhibit each other. So what happens when you push these closer is that the difference in response time between the two probabilities increases again. So opposite to before, it decreased it, now it increases again. And essentially it increases because you're making a selection decision. And in the selection decision, the competitive advantage between these two locations is reduced, or the difference between those in competitive advantage is reduced, because pushing them closer means both of them will be somewhat active, both have a chance to get activated. And therefore, it takes longer to move away from that common path, from the averaging path, and to move off to the selection tasks. It takes a longer time. So that is an account for the distance effect, for this classical distance effect. Okay, I have this last point about the time course of selection decision. I think I can actually do it. If you still have the energy, we'll go through this briefly, and then I can actually start with memory next week. So this was all measuring the classical reaction time. We used the fields to measure the predict the reaction time, and in these models, it's all just a question of how the preshape, which reflects the task set, the go signal, how strongly that overlaps with that, and the boost, how these three, the imperative signal is the specific input, and the go signal is the boost, how these three factors combine, which one dominates, and so on. And there's much more to that. If you look at that old paper, we modeled all kinds of effects like stimulus response compatibility effect, Simon effect, and so on, for those of you who know these sort of things. Now, this is not so exciting because it is very old stuff, and we have a new interpretation, the fact that these peaks have some neural significance, and that the metrics of these peaks have neural significance, and we predict metric effects, so that makes it a little bit more compelling. But on the other hand, we would want to perhaps really say what would be direct ways of observing the proposed spatiotemporally gradual specification of a state when the peak emerges. And it turned out that about at the same time there was activity at the same time when we started this, so it took us a decade to get this going. There was activity in Claude Gues's group in New York that spoke to this issue, even though they had actually different questions they had in mind. What they did was a paradigm that we have called the timed movement initiation paradigm. It goes back to someone called Brover in the 60s, but it was really Claude Gues's group that elaborated this paradigm. It's a paradigm where you're forcing participants to make a decision at a given moment in time. You give them a metronome. In one version, it's 4 different tones: beep, 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 beep. And the fourth one, you have learned in a familiarization session that's when you have to start to move. And you give only a manipulated interval before that fourth tone. You give the imperative stimulus. So this stimulus response interval is imposed from the outside. You can vary that. And that's by varying that, you can see the time course of the decision. People can learn this well enough so that they will actually generally make their response usually not right on the fourth tone. They make it a little bit delayed, but that delay is then roughly constant as you vary the stimulus response interval. It's only like 20, 30 milliseconds after the fourth tone. And here's another characterization of that. You see what happens. This is a typical task. They have a isometric force task. So you're controlling through a joystick the vertical position of some signal on the computer screen. The joystick doesn't actually move. It's just very stiff, and you're just pressing against it. And so initially you have some force level, and then you get a signal that tells you which of 6 force levels you can do. There are 3 forcing up and 3 force going down. This will be the target force you have here. And the solid line is what people actually do. You see when they hear these tones, beep, 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 the last one, they're supposed to move right here. You see they move, it's very good. They anticipate, and they really start moving the joystick right here. And then they actually only have to transiently hit the right target in this task. So they did this. It would be a correct trial here, where they did this correctly after a little time, a moment in time. And so what is varied experimentally is this time interval, how much before the fourth tone you're telling people through this display which of the 6 forces they have to produce. And that it's these 6 forces, that is something participants have learned across trials previously. So they know it's going to be up or down, it's going to be one of those 3 levels of amplitude. this is the sort of results you get. This is direction. So you can have up or down, and then in each direction there are 3 targets, the large amplitude, the middle amplitude, and the low amplitude. And to the right is time in the sense of the stimulus response interval. And so you see in this case, let's see, so in this case, the correct response was moving up to 50 Newton. And you see when you leave enough times, like 250, 300 to 400 milliseconds, most trials, they hit the right target. Similarly here, they have to go to the middle force level, and here they have to go to the bottom force level, all pretty correct. Actually, the 2 directions were put on top of each other by plotting it as a function of the correct direction and the wrong direction. So these would be the trials where participants went into the wrong direction. You see that that's only true for early trials. None of the slower trials with larger stimulus response time made that mistake. It's across all these conditions. The other thing you see is that there is within amplitude, there is a graded shift. So early on, you're sort of in the middle here, centered around the middle choice, very clear here, you know, it's sort of centered on the middle choice. Here it's centered at higher levels, here it's centered at lower levels relative to the target level. And then it shifts gradually to the correct choice. So for instance, here in this interval, you have values as the mean value that occur quite often that are never really required, like 40 newtons is not really something that's been ever taught, but participants move there. So you can see really a graded shift of the representation. That's true across the board. It's actually also true for these error trials. It's also interesting. So even though you're categorically off for direction, you're still making a convergence for amplitude, and because direction is bimodal, up or down, you know, you cannot conceptually even do that right. There could be different ways of plotting that to get that right, but there is a correct evolution in amplitude. So this speaks to the graded evolution that's continuous in time for movement preparation. You can literally observe in a way what's in the brain. So in the model, we can easily model that. So for instance, if these would be the trials with the short stimulus response interval, medium and long, and we've been that in some way, and then we're also looking at the peak forces being produced by the model, and the model just assumes that the axis of the field is peak force. We don't model the motor control part, how to actually do it. And the 3 choices, and you see for the center choice, we just have a sharpening of the distribution. For the top choice, we have this default response centered, you know, over the average of the 3 locations. The 3 lines are 3 through 3 force levels. Sorry, clicking here too much. And then you see the graded shifts, including a intermediate distribution that is centered on some intermediate value, and then you ultimately get to the correct choice. And you can compare that across theory and experiment. You can hardly tell which is which here. So we were able to account for that correctly. There was a whole bunch of different things that we were able to model that way. I want to point out at this last study, that was actual movement. A lot of these are isometric force pulses, which are very fast and easy from the motor control point of view. This was actual movement that's related very much to what Jean Requin saw in his study, because he used a very similar paradigm, where people moved to different targets and they were varied in terms of the angle between the directions and in the amplitude. There were 2 amplitudes. And you see, if you do this, then you vary here from the metric effect, right? From narrowly spaced choices to very broadly spaced choices. And then you also have time. You have fast trials, and stimulus response interval short. This is the thin solid line. I think it's thinly dotted. So it's this line here, thinly dotted. And then the intermediate times is this broadly dotted. And then the solid line is the long stimulus response times. And you see here, these are the 2 choices in terms of angular separation. And you see here that the early times are monomodal. It's an averaging solution. Just again, like I showed you earlier for saccadic movements. And then becomes broader at intermediate times and in the end becomes bimodal. It's become bimodal here because this mixes the trials where the leftmost was the choice and the trials where the rightmost was the choice. Of course, in different trials, it will be only 1/2 of that distribution. See that around 60-degree separation of the targets, you see a transition from the earliest really still being monomodal with a little side peak here, and then it becomes bimodal for larger times, and then for the largest times, it's clearly bimodal, that is, which actually means in an individual trial that you're on the correct side all the time. And then it stays like that for larger separations. So these authors actually theorized about different modes, discrete versus continuous mode, but it actually reflects the metrics of the underlying field. It reflects when the peaks split into two peaks, which reveals how broad they are. And if you look at the numbers, this is about the width where a cosine function of the kind that we see in the population code that was neurally estimated matches. I wonder if I have a slide about that. Maybe I don't. Maybe two down here? No, I don't. But you remember when we talked about the population representation of movement direction, the width of those peaks, that would be approximately consistent with that. I mean, it's not precise, and the width of those peaks also depend on the functions we use to fit the population distribution, and it's very crude resolution, so I'm not putting my hand on that. But there you remember that early peaks were reflected the average of the locations, and then they grow out at the correct location, and that would give rise in this picture to the bimodal peak, because on some trials it would grow out of the monomodal here, and sometimes grow out of the monomodal on the other side. So I mention this because it's sort of like measuring the width of the peaks early in the evolution of a motor plan, and then actually tracking that width and spatial location over time with different stimulus response intervals. So it's almost like making a measurement of the brain, making it behaviorally, and it's very interesting. And that can be modeled correctly. You see, in model, we can sort of see the transition from one peak to the other peak in the my model case. So I want to conclude here and point out that this DFT concept of selection decision through competitive interaction with a transition from averaging to selection is supported by a lot of behavioral signatures. I only looked at a small number, but there is a lot in quite different kinds of experiments that all converge on that picture. And essentially, these signatures emerge from the relationship between these three different things. The one I didn't listen is the imperative signal itself, the specific input. Oh, that is the specific input. The go signal is the boost, and the other is the third is the task set, the what we call the pre-shaping input. And the metric of the task layout matters, and we can understand the time course of decision making. So that concludes today's lecture, and we will be looking then next week at the role of memory. We'll look at sustained versus non-sustained and how that emerges, and we'll look at mechanisms for a memory taste. So two aspects of memory, working memory, and then a new aspect of learning. And I'll show you a study that combines all the effects that we've been discussing so far. So see you next week.